Matt explained. Nerve system or neurologic system is divided into two main divisions. The central nerve system, which is abbreviated as CNS, and the peripheral nerve system, which is abbreviated as PNS. The central nerve system consists of the brain and spinal. It consists of the brain and spinal cord. The peripheral nerve system contains the neurons that are not included in the central nerve system. And the nerve system consists of neurons and neuroglia or neuroglial cells. The neuron's main function is to transmit messages. The neuroglia main function to support and nourish nerve cells or neurons, okay? And neurons is the functional unit of the nerve system, and it is the basic unit. Today's main focus would be on neurons. Let's look at the neuron structure. So we have the structure of a typical neuron. A neuron is said to be typical when it has so many dendrites and one axon. The cell body, the dendrite, and the axon. The cell body contains the nucleus and organelles. It is the metabolic center of the cell. And the dendrite are some short, thick extensions from the cell body. Their main function is to receive signals and to receive changes, and then they transmit these messages toward the cell body. The axon is a long membrane-bound projection from the cell body. Its main function is to carry messages away from the cell body. The axon can be so long, some of them may reach to 1 meter. Each axon at the end branches to many fine endings, and at each ending we have a swelling called an axon bulb or an axon terminal. These bulbs store some vesicles that contain some chemical called neurotransmitter. These chemicals are important for the communication between neurons, okay? We will study them later on. And also, some axons, not all, have some lipid layers. Myelin sheath. The myelin sheath is an insulator, and an axon with myelin would carry messages faster, okay? So when we have myelin, the messages, the impulses would be transported faster. So there are some type of cells that produce myelin. There's a type of cells that produce a myelin in the brain, and there's another one that produce myelin for the neurons that are part of the peripheral nerve system, and these are called Schwann cells. So, the myelin sheath is formed by a type of neuroglia called Schwann cells. Schwann cells produce myelin for neurons that are part of the peripheral nerve system that do not make the spinal cord or the brain. They are part of the peripheral nerve system. Each Schwann cell contains a lipid substance called myelin in their plasma membrane. The myelin sheath develops when Schwann cells wrap themselves around an axon many times, and in this way lay down several layers of plasma membrane. If I magnified this part of the neuron and we look, you will see that this is a cell. This is a cell that surround axon. This is a cell that surrounds a part of an axon. It is nucleus. This is the plasma membrane. This is also layers of plasma membrane. Between these layers, we have our lipid substance called myelin. This is an axon. This is our cell called Schwann cells. This is the nucleus. This is the myelin. And it do what? It wraps itself around the axon many times, and it forms layers of this plasma. Between these layers is myelin. So that's why it is called myelin sheath. All of this myelin sheath. Mostly, the long axon contain myelin, and the short one do not contain myelin most of the time. Because the Schwann cell myelinate only a part of an axon, it myelinates only a part of the axon, the myelin would be interrupted and gaps will form between these cells. These gaps are called nodes of Ranvier. Now we looked at the structure of a neuron. Let's look at how neurons communicate and transmit messages. Let's draw a human. Let's try to magnify his hand. We will see that there is so many neurons there. Okay, so consider this is a muscle cell. If this human want to move his hand up, the message would be transported from his brain to the neuron in his hand, and this neuron would send this impulse to the muscle in order to contract and move. Messages move through the nerve system by two ways. First, within the neuron, the messages are transported as electrical impulse called action potential. And then between neurons, messages are transmitted chemically by a substance called neurotransmitter. I just looked at the internet and it says when messages move in the neuron, it means they're transported, and between neurons, they're transmitted. This is called action potential. Action potential only happen at beginning of the axon and ends at the ending. For neurotransmitter move to the next neuron, this neuron receive a signal. Between neurons, messages are transmitted chemically by a substance called neurotransmitter. Before talking about these ways, let's look at this cell, plasma membrane. And for example, this ion, these positive ions have a negative charge. They have a negative charge. And this blue ion has a positive charge. The number of positive ion inside is greater than the number of positive ions outside, and the number of blue ion inside is less than the number of blue ions outside. So what happens? We know that there's more negative ions inside the cell than there's outside, and there's more positive ions outside the cell than there's inside. The cell become negative charge, negative. The cell has a negative charge with respect to the exterior of the cell. Because there is difference in potential across the cell membrane, we will have voltage. And now the question is, does this ion move through the membrane? Yes, some are permeable. For example, this tiny pink ions is permeable too, so it can move through the cell membrane. Some ions are just so large, they cannot move through the cell membrane. They have some specific protein in which they pass through it. This protein acts as an ion channel. For example, this is an ion channel that is just for the large pink, and this is the blue ion channel that is specific for the blue ion. And some are closed. For example, here this is closed, these are open. This is closed, this is open. A neuron, like every other cell, has voltage or membrane potential. Voltage or membrane potential. There is ion inside the axon and ions outside. The concentration of these ions are not the same, and these ions have their own channels. Some of these channels are closed, some channels of other ions are open. We know that ion channels are protein. There are so many types of ion channels, and each ion channel is specific for a specific type of ion. For example, in neuron, we will look at potassium ion and sodium ions. Each channel is for a certain type of ion, or each channel is for a specific ion. For example, sodium channel only allows sodium ion to pass through it. If a neuron have myelin, the ion channel would be located at nodes of Ranvier. Ion channels located in nodes of Ranvier. And there are so many types of ion channels. And some of these protein ion channels open and close due to changes in voltage. So they will be called voltage gated ion channel. Other type of channel may open and close due to chemical like neurotransmitter. These are called chemical gated ion channel. And there's another type of ion channels. It's called leak channels. Sodium leak channels or potassium leak channels. But today I'm not going into these details. I will call all of them just ion channel. The nerve system is so complex and there are a lot of details in it. If you want me to explain things in detail, comment that. The nerve cell, a neuron, as we said, like every other cell, has a membrane potential. It has ions inside and outside. Their concentration is different. Each ion has its own ion channel. Some of these channels are open, others are closed. These, for example, sodium channels and potassium channels do not open and close at the same time. If the channels are open, these ions would tend to move due to the concentration. Our potassium channel is open. In which direction this potassium ion would move? They would move according to their concentration gradient. Now the concentration of potassium ion are higher inside and outside is lower, so these ions will tend to diffuse from area of higher concentration to an area of lower concentration. So the movement of ion is according to the concentration gradient. Let's look. I'm gonna talk about the potassium ion, K positive ion, the Na positive ion, sodium ions. We have another one is called the large negative protein, and they are located inside the eggs and they are just so large, so they just stay here. And I draw this picture according to the resting state. When a neuron is not receiving or sending a message, it is in a resting state. In the resting state, the concentration of potassium ion are higher inside than outside, and the concentration of sodium is higher outside than inside. The nature of a nerve impulse has been studied by using excised axons and a voltmeter called an oscilloscope. This is an axon. If we cut it, we call it excised axon. Oscilloscope, which is a voltmeter, it measures the voltage. The voltage is a measure of electrical potential difference between 2 points. And in this case, we have an axon and we are measuring the electrical potential difference between the 2 points. And these 2 points are the inside and outside the axon. When the axon is not conducting an impulse, the oscilloscope records a potential difference across a membrane equal to about negative 65 millivolts. This number indicates that the inside of the axon is negative and outside is positive. And this state is called resting potential. Resting potential. An axon is said to be at rest when it is not receiving or sending a message. When we use an oscilloscope to measure the potential difference across the membrane, it reads negative 65 millivolts. When an axon is at rest, it has a potential difference of negative 65 millivolts. When an axon is at rest, the inside is negative, the outside is positive. Voltage is often measured in millivolts. Here we have excised axon. This is axon membrane. We have used an oscilloscope to measure the potential difference across the membrane. And this axon is not receiving or sending an impulse. It is at rest. And when we use the oscilloscope, this device measures a potential difference of negative 65 millivolts. In some books, I have seen that it is negative 65. In other, it said the resting potential is negative 17 millivolts. The important thing to know, when the axon is at rest, the inside is negative and the outside is positive. The question now is why inside the axon is negative during the resting. We will find the answer by looking at the behavior of ions and their charge and the channel. So during the resting state, the concentration of potassium ions, these pink are potassium ions, the concentration of potassium ions inside the axon is higher and outside is lower, and the concentration of sodium ions inside is low and outside is high. There is also a high concentration of these large negative proteins inside the axon. So if we look at the ions profile that are inside the axon, the concentration of potassium is high inside, the concentration of sodium is low inside, and the concentration of these large negative proteins are high inside. And sodium and potassium both have positive charge. These proteins have a negative charge. The outside of the axon, the concentration of potassium is low, the concentration of sodium is high, and this is the ion profile during resting state. Some of potassium channels are open. These are called potassium leak channels, and potassium, which has a positive charge, tend to diffuse out of the axon because the concentration of potassium is lower outside. So things move from higher concentration to lower concentration, and things tend to diffuse from area of high concentration to an area of low concentration. And these large negative proteins stay inside the axon. These are just large negative protein, and they stay inside the axon because they're just too large and they cannot move to the outside. We have voltage gated ion channel. The voltage gated ion channel for sodium is closed, and the voltage gated channel for potassium is closed, and there's also leak channels. The membrane is permeable to sodium and potassium ion, but it is more permeable to potassium ions. So potassium tend to diffuse outside through the leak channel more than sodium ion diffusing. And if K positive, potassium moves to out, diffused out of the axon, and sodium ions moves to the inside or diffused into the axon, while their concentration do not become equal on both sides. Why? Now this is an axon. We have more potassium inside than outside, and we have more sodium outside than inside, and they have their leak channels, and the membrane is permeable to these ions. They can diffuse into and out of the axon. Why does not their concentration become equal? The same amount of potassium ion on both sides and the same amount of sodium ion on both sides. The unequal distribution of these ions is due to the action of sodium-potassium pump. Sodium-potassium pump is a protein membrane that actively transports potassium into the axon and sodium out of the axon. It do that by using ATP. I said actively. They tend to diffuse passively, but this one do the opposite. The sodium-potassium pump uses ATP in order to move sodium outside and to move potassium inside. So there would be a larger potassium inside and smaller potassium outside, a larger concentration of sodium outside and a smaller concentration of sodium inside. That is the goal of sodium-potassium pump. Because sodium-potassium pump moves potassium to their higher concentration site, it moves potassium into the cell and sodium out of the cell. This is before and this is after. The result is unequal distribution of ions, and this unequal distribution causes the inside of the axon to become negative with respect to the exterior. That is why at rest the oscilloscope reads a potential difference of negative 65 millivolts. The black one is before. We have larger concentration of potassium inside than outside, a larger concentration of sodium outside than inside. But the goal here, the goal of sodium-potassium pump is to maintain the unequal distribution, so it makes potassium concentration larger inside than outside and sodium larger outside than inside. but it does not become so much larger because they're already passively transporting. This do the reverse process. And now we talked about the resting state of the axon, the resting potential, which means the inside is negative, and why the inside is negative. The inside is negative because there is large negative protein inside the axon, and this sodium-potassium pump make an unequal distribution of ion. Okay, just to review that, we have an axon during resting. We'll have more potassium ions inside than outside and more sodium ions outside than inside. And the voltage gated channels are closed, but there are leak channels, and these ions tend to diffuse from area of higher concentration to an area of lower concentration. But the thing is that the membrane is more permeable to potassium ions than to sodium ions. Okay, so potassium ion would be moved outside more than sodium ion entering the axon. And also, sodium-potassium pump will try to maintain the unequal concentration, so it moves the potassium into the cell again, and it moves the sodium outside the cell again. So this is active transport, and the black arrows show passive transport. So there would be more positive ions outside the cell than inside, and the inside would become negative with respect to exterior. Inside is negative not because there is no positive ion inside, but because there are more positive ions outside than there is positive ions inside the cell. Okay, and we also have some negatively charged protein, few number of positive ions and a large number of positive ions outside. The inside becomes negative. Sodium-potassium pump have important role in maintaining the resting potential, and after an action potential ends, it restores the resting potential. When a neuron receives signal from a neurotransmitter, from an excitatory neurotransmitter, or it receives a stimulus, if these things are strong enough to cause a change in membrane potential from negative 65 millivolt to about negative 14 millivolt, an action potential will start at the tip of an axon, and it would move along the length of the axon until it reaches the axon bulb or axon terminal. And from negative 65 millivolt, negative 40 millivolt, negative 40 millivolt indicate that the inside is negative with respect to the exterior, but it is less negative than before. Okay, so let's look, and this number is called the threshold. So if a stimulus causes the axon membrane to depolarize to a certain level, that is called threshold. Let's look at this. Action potential will start at the tip of an axon and end at axon terminal. Let's look at the action potential definition. Action potential is a rapid change in polarity across an axon membrane as the nerve impulse occurs. Action potential is an all or none phenomenon. And that means that the strength of an action potential does not change, but the frequency changes. If we have an intense stimulus, this intense stimulus would cause an axon to fire more often in a given time interval. It would cause a neuron to generate an action potential, and then another one, and then another one. But a weak stimulus may not cause an action potential, and if it is not that weak, if it causes an action potential, a neuron may generate a single one or just few of them in a given time interval. Okay, so the strength of an action potential does not change as the strength of the stimulus changes, but the frequency of the action potential changes as the stimulus strength changes. Fire here means action potential. Action potential goes segment by segment. Now, first action potential will start at the tip of an axon, then this segment would go through action potential, then this segment, then this one, then this one, then this one, until it reaches the end and causes the release of neurotransmitters. First segment goes through action potential, then the next segment goes through it. This one would finish its action potential, then this segment would go through it, and this one would finish its action potential, then this segment would go through it, and this would finish its action potential. This segment would go into action potential, and this one would finish, and so on. It goes segment by segment, and this process ensures that the action potential moves in only one direction, and that is away from the cell body and toward the axon terminal. And action potential are self-propagating. Each action potential generates another along the length of an axon. As we say, this segments, then this one, then this one, then this one, then this one, and so on. And today I will just explain how action potential happen at segment 1 in a time interval from the beginning to the end, and other segments, they just repeat the same process. An action potential travel across the length of an axon segment by segment. Now first we have segment A, which is at rest, and segment B is at rest. When an action potential starts at the tip of an axon, segment A goes through depolarization process and the inside becomes positive. We know that during rest the inside is negative, but during an action potential the inside becomes positive. And this stage of action potential is called depolarization. And then it would become negative again, and this stage is called repolarization. And this means it is going back to rest. And the next segment would start at its action potential and would go through depolarization. But the thing is, repolarization of the first segment happened before the depolarization of the next segment. And then the first segment goes through the refractory period, which is a state in which a segment cannot receive a new action potential because sodium-gated ion channels are closed, and this prevents the previous action potential from going back. So this is the action potential for the segment A, action potential for segment B. All segments go through these, but not at the same time. The first segment would go through these, then the second one, then the third, then the fourth, and until it reaches the end. First, we have an action potential which consists of depolarization and repolarization, then the refractory period, and then the rest. And for action potential, we'll look at potassium ions, sodium ions, their channels, their gated channels, and their concentration and their movement. And we will not look at the negative protein because they stay inside the axon. We will look at a single segment, then all the other segments would repeat the same process. Now I draw this, and let's look at this segment, this first segment. And when an action potential happens at the tip of an axon, what happens inside? What happens at a microscopic level? Now this is the voltage. When a stimulus that is so strong that causes a depolarization to about negative 40 millivolts, the inside would be less negative than the resting potential. During depolarization, sodium-gated channels open and sodium ions rush into the cell. And by that, the inside become more positive with respect to the exterior. The inside become positive, the outside become negative because there are more positive ions. There are a lot of potassium and sodium ions inside than there is outside. Outside become negative with respect to the interior, and interior become positive with respect to exterior. During depolarization, the voltage changed to positive 40Y. Sodium-gated channels open and sodium ion rush into the cell, so there will be a large number of potassium ion inside, large number of sodium ion inside. Inside become more positive until it reaches positive 40 millivolt. During depolarization, potassium-gated channels are closed and sodium-gated channels open. Remember that during resting, potassium concentration was high inside and low outside, sodium high outside, low inside, but during depolarization, gated channels for sodium open and sodium rush into the cell. So the result is there are a lot of potassium ions and sodium ion inside than there is outside. Potassium cannot move outside during this time because its gated channels are closed. Potassium cannot move to outside during depolarization. During depolarization, Na positive gated channels open and sodium ion rush into the axon, making the inside positive with respect to the exterior. Because Na and both have a high concentration inside, so there is more positive ion inside become positive. And then repolarization stage begins, and that is when potassium gated channels open and sodium gated channels close. And the result is that potassium ion rush out of the axon, making inside negative with respect to the exterior from positive 40 millivolt to negative 40 millivolt. Here is the repolarization. Sodium gated channels close and potassium gated channels open and potassium rush out of the cell. The inside becomes negative again. Depolarization and repolarization make action potential. So action potential consists of two parts. A part where the inside was positive and the part where the inside was negative. This is outside. This is rest. The resting potential inside was negative. Action potential consists of two parts where the inside is positive and when the inside is negative. Inside is positive, it is depolarization, and inside is negative as repolarization. And it ends when it reaches negative 40 millivolt. Here what happens, potassium gated channels open and potassium ions rush out of the cell. Here what happens, potassium gate channels open and potassium ion rush out of the cell. So here what happens, we have large concentration of Na, small outside, but a large concentration of potassium that is going to outside. And here is small. And the next segment would start at a depolarization process slightly after the repolarization of the first segment. And again, like the first segment's depolarization, sodium gated channels open and sodium ion rush into the cell, making inside more positive, outside negative. Look, during action potential of the first segment positive, the inside is positive, outside is negative, and the second segment is at rest. Then the action potential goes to the next segment. The next segment becomes positive, outside negative. This is repolarization. This is also action potential negative, positive. And that's how action potential goes. And each preceding portion causes an action potential in the next portion of an axon. As soon as an action potential has moved on, the previous portion of an action undergoes a refractory period. A refractory period is a state at which the previous segment cannot generate an action potential. During this period, sodium gates are unable to open. This ensures that the action potential cannot move backward and instead it moves down an axon toward its branches. And this is where a refractory period happens. And this is where a refractory period happens. In this stage, the membrane is more negative than negative 65. And that is because there are a lot of potassium outside and a lot of sodium inside, and that is not the resting state. The resting state should be like this, more potassium inside and more sodium outside. So in order to move from here to here and from less than negative 65 to negative 65 millivolt, we need the action of sodium-potassium pump. Here sodium-potassium pump work. It moves potassium inside and moves sodium outside by using ATP. So this is after an action potential. Potassium concentration is higher outside and sodium is higher inside. sodium-potassium pump restore the resting state by moving potassium inside and sodium outside, and so the voltage would change from less than -65 to -65 millivolt. Less than, it may be -75, -80, and so on. And now the resting potential is restored. Let's summarize. Depolarization is the first part of action potential in which inside is positive, outside becomes negative. Potassium concentration, this is inside axon, is high. Sodium concentration become high because it is entering. And what happens? We have two voltage gated ion channels. Potassium voltage gated ion channel and sodium voltage gated ion channel. During the depolarization state, potassium voltage gated ion channels are closed, but sodium voltage gated ion channels open and sodium ions rush into the cell. During repolarization, inside the second part of action potential, inside become negative, outside positive. Why? Because potassium channels open, sodium voltage gated channels close, and potassium rush out of the cell. So inside become negative. So potassium concentration inside the axon would be less than before. Sodium concentration would be high, and refractory period, the inside is negative, outside is positive again, and we have a lot of potassium outside, a lot of Na inside. So inside axon, potassium concentration is low, sodium is high, and the sodium-potassium pump moves potassium inside and sodium outside. Neurons communicate with other neurons at special junctions called synapses. A synapse consists of a presynaptic neuron, a synaptic left, and a postsynaptic cell. The cell may be a muscle cell, it may be a neuron, or a gland cell. And when action potential reaches the axon terminal, it causes the release of some chemicals called neurotransmitter. These neurotransmitters travel across the synaptic left to the postsynaptic neuron, and this neurotransmitter may cause this neuron to start a new action potential or to inhibit. So neurotransmitters have inhibitory and excitatory effects on the postsynaptic cell. It depends on the type of neurotransmitter and on the channel to which bind to it. When an action potential reaches the axon terminal, it causes the calcium ions to enter, and these vesicles, which enclose neurotransmitter, fuse with the presynaptic membrane, presynaptic neurons membrane, and it causes the release of neurotransmitter. The neurotransmitter move through the synaptic left and they bind to these receptors. But neurotransmitter just bind to the receptor. They do not enter the neuron. It causes sodium ions to enter the neuron, and this causes the generation of action potential in postsynaptic neuron.